So at AACR this year, I'm presenting some results from the TATTON phase 1 study. This is a large multi-arm study looking at different combination drugs for EGFR patients who have acquired resistance. And what I'm presenting is results about the Savolitinib combination, a MET inhibitor, together with osimertinib, third generation EGFR inhibitor. And we ran 2 different expansion arms in this patient population, one for EGFR patients who had developed MET-driven resistance while on a first or second generation EGFR drug, and then the second cohort was for EGFR patients who had developed MET-driven resistance while on a third generation EGFR drug. And both cohorts were treated with osimertinib and savolitinib. The primary endpoint was looking at the safety. Overall, the combination had a little bit more toxicity than we're used to seeing with osimertinib alone, but it was manageable in the majority of patients. A little bit of extra GI toxicity was the most commonly seen, nausea, vomiting, as well as fatigue. Response rates were quite high in this biomarker selected group. For the patient cohort that had developed MET on a first or second gen drug, the objective clinical response rate was 52%, and the duration of response was approximately 7 months. For the more heavily pretreated patients who had developed MET-driven resistance while on a third generation drug, the response rate was about 25%, with a median duration of response around 9 months. So overall, this is one of the first studies to show that you can successfully treat with a targeted therapy EGFR patients who develop acquired resistance driven by MET. And we need some further studies to gain more clarity around the type of responses and the type of MET that is most predictive of those responses. And some studies will be ongoing this year. So what's interesting about the cohort of patients who had received a prior third generation inhibitor is that over 75% of that group had received osimertinib already as their third generation inhibitor, and now are responding to a combo of OC plus savolitinib. We know from preclinical work that when the EGFR resistance is driven by MET amplification, you probably cannot successfully treat that alone with only a MET inhibitor. You still need the EGFR inhibitor because underneath it all, the cell is still addicted to EGFR. And so the combination aspect, I think, is quite important. So the TATTON study had a lot of different patients enrolled from around the world, and the cohorts were defined specifically so that we could have better hypothesis generating, better clarity about what is the next group of patients to study. If we had lumped all the EGFR patients together in one cohort, it would be a bit difficult to tell whether the different groups were responding. Some would be receiving osimertinib for the first time when they got the OC-Savo combination. Some had previously progressed on osimertinib and then were getting the combination, and a variety of different number of lines of prior therapy. So separating the groups into these 2 cohorts prospectively, I think really allowed our study team to see what the signal was coming from each individual group of patients. So the osimertinib and savolitinib combination will be studied in a couple of phase 2 settings to try and get more information about the effectiveness of the combination. The Savannah study is already open and enrolling patients, and this is a single-arm study of OC and Savo in patients who have received osimertinib in the past, whether it's in the first, second, or third line setting. And then the Orchard study is going to be opening later in 2019. That's going to be examining specifically first-line OC progressors who then develop MET as their resistance mechanism. I think this is something that we're going to be seeing a lot more now that OC has moved into the front line, and MET amplification makes up a pretty decent group of the resistance to osimertinib. So those patients will be able to enter the Orchard study.